So my dear children, in the earlier chapter we discussed the first and foremost method which a transportation process is going to happen within a plant. So that method was the diffusion. So diffusion means in simple transportation or traveling of particles from higher concentrated area to a lower concentrated area. That's it. Right? From a place that has high number of particles to a place that has less number of particles. Automatically these particles are going to travel. This is the process which is named as, or which was named as, the diffusion process. And we discussed several examples where the diffusion can be seen in a plant, like the absorption of carbon dioxide needed for photosynthesis, releasing of oxygen gas after the photosynthesis process, absorption of oxygen gas for the respiration process, and removal of carbon dioxide and water vapor after the respiration process in plant. So these are the special instances that we can observe the process of diffusion, the main transportation method, the first transportation method in a plant. Now my dear children, we are going to discuss another transportation method, another main type of a transportation method that's going to occur within plant. So this method is referred as the osmosis. Osmosis. So osmosis is the second method of transportation. Way much similar to the diffusion process. Actually, it is also a type of a diffusion. But however, there is a small bit of a difference in here. Okay, small bit of a difference. Same way, the diffusion is the one which is going to occur, but a small bit with a small bit of a alternation. That's it. Okay. So now let's see what is this osmosis is. Okay. So my dear children, the process of osmosis. Before we are going to discuss about the osmotic process or how or what this osmosis is, you are given with an assignment. So this assignment is really simple. What you have to do here is uproot a balsam plant carefully without breaking its roots. So balsam is a flower, right? It doesn't have a woody stem, right? Mainly the stem is like stem is like mainly like a liquid kind of a one, right? Doesn't have a woody stem, balsam, right? And there are several colors of flowers could be seen. Balsam plant, right? So you have to uproot a balsam plant without breaking the root system of it, because the root system is very important in this experiment, my dear children. Okay. So the root system is very important. So when you are uprooting this balsam plant, make sure to keep the root system within the plant itself. So carefully do this thing. Okay, at your home you can do this thing, right? You only need to find out the balsam plant. Then wash soil in the roots. So we have to wash it very well as there are soil, right? So we have to remove that soil and dirt from the roots. Put some water into a container and dissolve some red ink in it. So we have to put some water and we need to dissolve red ink in it. Okay, need to dissolve some red ink. Now dip the plant in the water you prepared with ink. Observe after few hours. Now what you have to do? You have to dip the plant in ink, right? Ink with water, and you have to leave this plant for about few hours in that water. Okay. Then after few hours, you need to observe what's going to happen here. So you know that, my dear children, plant roots are the ones which is going to absorb water. Plant roots are the ones which is going to absorb water from the soil. So we have uprooted here a balsam plant, right? And the specialty in here is that the root system is still within the plant right now. Okay, root system is still within the plant. So when I'm going to place it in this red ink containing water, definitely water should be absorbed by the root system of the plant. Okay, that's the thing which is going to occur here. As there are red ink as well, we can clearly observe the red color nature along the root of the plant. And if that water is going to transport to different places other than the roots, then also we can observe that unique red color in it. Okay, I mean, like if that water is going to transport towards the stem, towards the other places, then we can definitely observe that red color. From the outside we can see it, right? From outside we can see that red color. So my dear children, now within this activity we'll be doing that thing in order to see whether the water particles are going to transport from one place to another through cells. Okay, you know that each and every organism contains cells. Plants contain cells. So root hairs, they contain cells. So there should be a way that the water is going to pass from one cell to another. That's how this process is going to happen, right? Otherwise there can't be any traveling method, no? There are no like tubing systems in the plant, no? So water should travel from one particular cell to another one. That's how the water should travel in here. If water going to travel in like that way, we can observe that red color, my dear children. Okay. So water is getting absorbed from the roots. If it is going to travel towards the stem, then we can see that unique red color as there is red ink. Okay. So now we are going to see whether this thing is going to happen or not. So now I'm going to prepare. I'm going to go to my lab and I'm going to prepare this setup and I'm going to observe whether this thing is going to happen or not. Okay, so my dear children, I'm going to take you to the laboratory. I'm going to prepare this setup and I'm going to show you that you can observe that red color, that unique red color within the stem of the plant, stem of the balsam plant. Okay, so let's head on to laboratory and we'll conduct this activity, this assignment, and we'll see whether the expected observations could be taken or not. Now, my dear children, I have a balsam plant with me. Now you can see that the trunk or the stem of the balsam plant is having a green color, right? It's having a green color. So what I'm going to do here is I'm going to take this balsam plant and insert this thing into the potassium permanganate solution or to the Condis solution. So I have inserted it. Now we need to keep this thing for a while so that the roots will absorb this solution very well. Then after that, let's see what's going to happen, right? So let's keep the system for a while. Then after that, we'll observe what's going to happen in the next step. So my dear children, within this activity, we'll be observing the nature of osmosis. So in order to observe the nature of osmosis, I have with me a colored water setup. Now in this beaker, you can observe red colored water, right? And I have several balsam plants, right? I have several balsam plants. So as you can see here, the trunk or the stem of the balsam plant is in greenish color right now. Okay, it's in greenish color. And I have somewhat a smaller one as well in here. Now what I'm going to do is I'm going to insert these balsam plants into the colored water solution like this. Then after, we have to observe Then after we have to observe the two plants after like 2 or 3 hours. So let's keep the system in the same position for about 2 to 3 hours, and after that we'll observe what's going to happen or what's the change within the stem in the plant, right? So we'll leave it for some while and let's observe after some time. So my dear children, now I have kept this balsam plant for some time at this colored water. Now you can clearly observe the color change. Now the earlier this plant was in green color, greenish color. Now you can observe at the base of the plant the red color is very well observed, and at the top of the plant also there are few colors, right? Few, I mean like red color can be identified in few stops, right? So here, at here and over here also you can observe that red color very well. And within the base, like in this area, the red color is quite well observed. Now it's because that root hairs contained within the balsam plant is going to absorb this water that has red color pigments, and it's been transported towards the xylem tissue, right? So that xylem tissue now in red color because of the red color pigments. So that's the action of osmosis process. So here the root hairs, the membranes in the root hairs is acting as a semi-permeable membrane. So it allows to flow water through the membrane. So by that way the water is going upward through the balsam plant. So that's why we can observe the red color in here, right? So my dear children, let's move on to the classroom once again and we'll write down the observation and the conclusion of this experiment. Right, my dear children, now you could clearly observe that the color of the stem has got changed dramatically to red color over there because of the red color ink absorbed from the plant. Okay, actually here the water was absorbed by the plant root and they travel from cell to another cell and through that to the stem. There is a clear upward motion of the red color water. So by looking at the stem of the plant, we could observe that red color because the water particles has traveled from one particular position to another through the cells within the plant. Here you know that plant is going to transport water and minerals through a special tissue which is referred as xylem. Xylem tissue is the tissue that can transport water and minerals. So root hairs are going to absorb water to a cell, and from that it is being transported to the other different cells, and from those cells to the xylem tissue. Then xylem tissue is going to transport water all around the plant. So here we kept only for few hours, no? So that's why we can clearly observe the red color on the stem. If I'm going to keep it like for more time period, then the whole plant would get into red color because of transporting red color water. So my dear children, the observation is given here. See, you will see that the roots absorb red ink solution and it goes up the stem. There is a mechanism that water particles and ink particles travel through root cells and enter the xylem tissue. So like I told earlier, there is some special method which the water being transported from root cells, from one root cell to another, and from that one to another, and to the xylem tissue. Okay, First of all, water should travel from one cell to another and then after to the xylem tissue. Okay. So my dear children, root hairs, okay, root hair cells is the one which is going to absorb water. However, that water should travel to xylem tissue. So definitely there is a mechanism that water particles travel from cell to cell. There should be a mechanism. There should be a special process where water travel from one cell to another within the plant. Here especially water, I am talking about water. Right? There should be a special method of transporting water from one cell to another cell. There should be a speciality. There should be a special method. There should be a special process in transporting water from one particular cell to another one, from that one to another one, then to the xylem. So my dear children, here we are investigating how this process is going to work. Okay, how this process is going to work. So to get some idea, we did a small bit of an assignment over here by using that balsam plant, and you could see that root hairs are definitely passing water from one cell to another, then to the xylem to transport water to the upper part of the plant. So now let's investigate further things related for this process. So you are given with a certain type of an activity, a special kind of an activity. In order to further investigate about this transportation process, which is going to happen from one particular cell to another. So here we are going to study how water particles travel from cell to cell. Right? How we are going to, down here we are going to specially study how these water particles are going to travel from cell to cell in a plant. So for this process, we'll be doing this experiment given here. It's a really simple experiment, right? We'll see what are the materials that we need. First of all, we'll be needing a thistle funnel. Thistle funnel. Thistle funnel is a special kind of a funnel that we use to pour substances like liquid substances into a certain beaker, right? So thistle funnel is very important. Like if you have a long neck beaker, then when you are pouring down like acidic substances or acids, the acids may spill here and there due to that long neck thing. So in that case, thistle funnels are used. It avoids that spilling part. Okay. So thistle funnel has a longer neck like this. It can be inserted to the longer neck beaker quite easily, so that spilling would be reduced. Thistle funnel, right? Thistle funnel. Thistle funnel. Then in the next one, we'll be needing cellophane paper. Cellophane paper, right? Not a polythene, cellophane paper. Okay, we'll be needing a cellophane paper. Then a beaker, rubber bands, we'll be needing some salt solution, right? Salt solution. Then water, then if you want, you can use a water throw also. Okay, water throw. Then condensed solution. Right, condensed solution. Right, so these are the materials that we need. We'll be needing a thistle funnel, cellophane paper, beaker or else a water throw. Right, if you can find out a water throw, then beaker is not important. If you are going to use a beaker, then beaker with some water. Okay, rubber band, like in order to tighten down that cellophane paper, right, we'll be needing several rubber bands. Then salt solution. Then after that, condensed solution. These are the main things that we need in doing this experiment. Right, my dear children? So let's now observe how to conduct this activity, or what's the method in doing this activity. So the method is given: fill the beaker with water. Now you need to fill the beaker with water. Cover the mouth of the funnel with the piece of cellophane. So by using the cellophane, you have to cover the mouth of the thistle funnel. Okay, you need to cover the mouth of the thistle funnel by using the cellophane layer. Okay, then place the funnel in the beaker and put condensed solution and salt solution into the funnel. Alright, so what you have to do, you have to insert condensed solution plus the water plus the salt solution into the thistle funnel like this. Now inside the thistle funnel, you can observe that quite unique purple color because of the presence of condensed. And into that system, you have to include the salt solution as well. Okay, you have to insert both the salt solution and the condensed into the thistle funnel. So here you can observe salt solution with condensed within the thistle funnel. Okay, then mark the starting level in the thistle funnel. Now you have to mark the starting position. Now this is the starting position. No, this is the starting position of the water level. Water level means the solution level within the thistle funnel. Starting position. So now we have to dip that thistle funnel within the water throw or within that water beaker according to the given figure in here. Cellophane paper is, or cellophane layer is tightly fixed to the mouth of the thistle funnel using rubber bands. Okay, it's been tightly fixed. Right. Mark the starting level in the thistle funnel. Note your observations after few minutes. Actually, we have to keep this system for a few time or for few hours. And after that, we need to observe what's going to happen. Right, my dear children? So this is the way of conducting our activity. Remember to mark out the starting position, the initial position. Okay. And make sure that you have tightly fixed the cellophane paper to the mouth of the thistle funnel. Right. So you add salt solution plus the condensed solution into the thistle funnel. Fix the cellophane paper very well, tightly to the mouth of the funnel. Then after that, fill the water beaker with water. Insert the thistle funnel according to the given figure. Then keep the system for about few hours and observe what's going to happen. Okay. Right then. So this is the way of doing our experiment, my dear children. Let's head on to the lab and we'll conduct this experiment. We'll set up this apparatus and let's see what's going to happen for this system after few hours. Now, my dear children, let's further investigate about this osmosis process. Now I have used here a thistle funnel fixed to the stand over here, and here I have placed a beaker. Now within the thistle funnel you can observe the Condis solution, right? Potassium permanganate or Condis solution. Now the Condis solution is having some amount of salt in it. So what I'm going to do is I'm going to pour this water into this beaker right now. Before that, my dear children, please check the level. So the level is now the water level or the solution level is now at here. It fills the bulb of the thistle funnel, right? Up to the bulb of the thistle funnel. Now you can observe the Condis solution. Okay, Condis with salt solution, right? Up to the bulb level you can observe it. This right up to the bulb level only. Now what I'm going to do is I'm going to gently pour this water into the beaker. Right. Now let's keep this system for a while and let's observe what's going to happen. At the bottom of the thistle funnel, my dear children, there is a cellophane paper, right? Cellophane paper which is firmly fixed to the mouth of the thistle funnel by using rubber bands. Okay. So if there is an entry or exit between the particles, that's going to happen only through the cellophane paper. It's going to happen only through the cellophane paper. So let's observe what's going to happen after some time. So remember, the water level was at the bulb level in the thistle funnel, not the water level, the Condis level or the Condis solution with salt. So that level was at the bulb level. Okay, it's exactly going to fill the bulb. So I have inserted some water. Now you can see the two levels of the water plus the solution in here, the Condis solution level. So let's keep the setup for several hours and after that we'll observe the changes. Okay, my dear children, now you can observe the setup very carefully. If you observe the setup very carefully, you can see that the Condis level, now inside I told you that we inserted Condis with salt. So here that level, the solution level has raised somewhat up. In the earlier case, when I was inserting the solution, I was inserting only to the bubble level. That's it. But however, now it's been raised up to some certain height along the thin tube. And if you check clearly the color of the solution, so the color of the solution has also get reduced, right? Earlier it had a very good purple color, but however now the color has got reduced somewhat. Reason for this thing is that inside the beaker, inside this beaker there is large number of water particles, but however within the thistle funnel there is less number of water particles as there are more salt and Condis. So automatically what happens here is water particles is going to move through the cellophane paper towards the thistle funnel. Okay, that's what's going to happen in here. So my dear children, let's head on to the once again to our classroom and we'll identify the further things related with our lesson. Right, my dear children, now we conducted the activity and you could clearly observe what has happened over there, right? It's like somewhat tricky, like we even though we have sealed the bottom part by applying that rubber band very well, however there is a method of transportation which has happened, Actually, water contained within the beaker now has traveled towards the thistle funnel. And you could observe it very clearly. Right? You could observe it very clearly. So in our observation before, now in this given figure, you can observe both the cases. Before it was like this, and after few hours it was like this. So here we can take two main very clear observations. First one, the water level of the thistle funnel rose up. Clearly visible to us. It was very clear to us that the water level within the thistle funnel has got increased. There is no any other possible way to increase the water level other than if there is a transportation or if there is a traveling of water particles from this beaker towards the thistle funnel. But from what kind of a place? Or through what? Water is not going to travel through glasses, no? So there is one possible way of traveling. Through the mouth. Through the cellophane paper. So my dear children, water automatically travels towards the thistle funnel, through the mouth of thistle funnel, through the cellophane paper which is being fixed to the mouth. Okay? Through the cellophane paper, the particles are going to travel. What particles? Water particles. Okay? So that is what has happened in here. So my dear children, the first observation, very simple, level of the liquid in the funnel rose up. So how can you say that the water has entered to the system? Simple. Earlier, the color of condis has a dark purple nature as there are a large number of condis particles. But by looking at these two figures, and when we are looking at the experiment also, observations of the experiment also, you could clearly see that the color of the condis solution has got decreased or reduced because the water particles has entered to the system through the cellophane paper. So as water is getting inserted into the thistle funnel, color of the condis solution is getting reduced as the concentration is getting low, or less as the number of particles of condis is getting reduced. More water, less condis. Therefore, the color is getting faded. Okay. So the second observation is level in the liquid level in the funnel rose up and the color of the condis faded. By that way, we can clearly say that water has entered the system, because there is no other possible way to reduce the color of condis. Okay. So water has entered to the system from the beaker. So therefore the level rose up. As water is getting inserted into the system, color of the condis solution reduced gradually. Okay. So this is our observation. So my dear children, it's clear to us that when I was conducting this activity and when I'm going to take my observations, during this observation, during this practical, we can clearly say that water is going to travel through the cellophane paper towards the thistle funnel. Through the cellophane paper, it's going to travel up, right? It's going to travel up. So this is a special kind of a mechanism. Water is traveling through some kind of a layer to a different place. Okay. through a layer to a different place. Right then. So my dear children, conclusion given. Now I am going to explain how this process is going to happen. Conclusion, right? So I told you that it has traveled from a certain layer. That layer is in here, the cellophane layer. Through the cellophane layer or through the cellophane paper, it has traveled towards the thistle funnel. Now I am going to explain what has happened. Okay, so let's go for the explanation then. The concentration of water molecules is higher in the beaker compared to the solution in the thistle funnel. Obvious, right? Because within the thistle funnel there was salt and there was condensed particles. Plus there was water also, but however, compared with the beaker, now within the beaker there is only water. Only water means 100% within the beaker we can observe only water. So the concentration of water particles within the beaker is very high. So obvious. Concentration of the water particles within the beaker is very high. Okay, there are large number of water particles within the beaker. There was no condensed particles within the beaker. There was no salt solution within the beaker, so therefore no salt particles within the beaker, only water. So the concentration of water within the beaker is very high, as there are very large number of water particles within the beaker. But however, if you take the thistle funnel has less number of water particles. So there is a concentration difference, my dear children. Definitely there is a concentration difference. When you are comparing the concentration of water particles, okay, there is a there is a huge concentration difference. Then there is a concentration difference between the condensed particles and salt particles as well. No, of course there is, right? Of course. Within the thistle funnel, condensed particles are very high. In the beaker, no condensed particles as well. So there is a concentration difference between condensed particles as well. Of course you are right. Within the thistle funnel, there is high number of sodium chloride or salt particles. Inside the beaker, less. So there is a concentration difference between sodium chloride or salt as well. But however, only water has been transported. No, only water has traveled. The movement is only observed in water. How can you say that? Because there was no color change in the beaker. And if there is a movement in particles from thistle funnel to the beaker, then the water level or the solution level in the thistle funnel would never increase. It would definitely decrease. So within this activity, the movement can only be seen in water only. The movement cannot be observed for condensed particles or for salt solution. Both remain within both of those particles going to remain within the thistle funnel. But however, water is going to travel from beaker through the cellophane paper into the thistle funnel, increasing the level of the thistle funnel. So here, even though there is a considerable concentration difference between condensed and salt solutions, these two particles, condensed or salt, they don't travel at all, but however, water has traveled. That means Within this process, only a selected particle can travel from one place to another upon a concentration difference. Okay, upon a concentration difference, only a selected particle can travel. Condensed never travel, salt never travels, only water travels. Means water is the only particle medium that can travel. Water is the only selected medium that can travel through the cellophane paper. Not condensed, nor the salt solution. So my dear children, see, the concentration of water molecules is higher in the beaker compared to the solution in the thistle funnel. So water molecules tend to move from area of higher concentration, which is the beaker, into those of its lower concentration, which is the thistle funnel, across the cellophane. So it's obvious to us that in here, water is going to travel from a higher concentrated area to a lower concentrated area in the same way like in the earlier process, which is referred as the diffusion. But however, other particles are not going to travel. Condensed particles are not going to travel. If so, there should be a color change within the beaker as well. But we couldn't observe any kind of color change within the water inside the beaker, right? So my dear children, only water has the ability to travel from a higher concentrated area to a lower concentrated area. Okay, so cellophane allows only water molecules. See, cellophane allows only water molecules to pass across and prevents salt and condensed molecules from passing across it. Now it's a really important process because cellophane can select what is the particle that it allows to move from one place to another through it. Cellophane has that ability. That cellophane layer or cellophane paper is going to allow only water to travel. That's it. So here, such membranes are called as semi-permeable membranes. Now my dear children, like these membranes are referred as semi-permeable membranes. Semi-permeable membranes. Semi-permeable membrane means it is a special kind of a membrane or else a layer that allows to pass only selected particles. Only selected particles. Now in this activity, within this activity, cellophane layer allows to pass water particles only. Condensed particles and salt is not getting passed through the cellophane paper. Only water is being allowed to pass. That's it. So only selected particles can pass through it. So therefore, these are called as semi-permeable membranes. Semi-permeable membranes. Actually, membranes or layers or these papers are in three types. Any kind of a membrane, any kind of a layer are in three types. We can divide membranes or layers into three types. These three types are impermeable membrane, permeable membrane, semi-permeable membrane. What do you mean by an impermeable membrane? Impermeable membrane means now there are two sides. You can see within this figure there are two sides. I'll mark these sides as A and B in each figure. A and B. Okay. A and B. between A and B in the first figure, between A and B in the first figure over here, you can see that there is a layer. And my dear children, this layer is completely separate in A and B into two parts. At the A side, there are different particles like this. There are different particles. Okay, there are different particles like this. So, as the layer in here is completely covered, these particles cannot enter to system B because it is entirely closed up. There is no way of entry and exit between the particles in here. So these layers are referred as, layer in A and B are referred as, layer within the A and B is referred as impermeable membranes, because these membranes would never allow any kind of a particle to pass through it. Impermeable, like polythene, right? Polythene, balloon membrane. These membranes, balloon membranes, polythene, they would never pass any kind of a substance through it. Not even water can't go through it. The impervious to water, balloon and polythene membranes. Cellophane is not like that. Cellophane is allowing water to pass through it. Okay? However, polythene is not. Polythene is not allowing to pass any kind of a substance through it. You know that you can keep, like, you can take a polythene bag and you can pour water into it if there are no any holes in it. Okay? You can pour water and you can carry it here and there. Polythene bag. It's because that water can't exit through the polythene bag to the outer environment. Okay? Because water is not allowing to pass any kind of a substance through it. So polythene, water can't, not water, polythene can't, polythene is not going to pass, or polythene is not allowing to pass any kind of a substance through it. So water cannot go through polythene particles because polythene is an impermeable layer. Polythene is an impermeable layer, right? Water can't go through it, but however, cellophane is not. Water can easily go here and there through cellophane paper. So my dear children, polythene is an impermeable layer, however, cellophane is not, right? So impermeable means particles cannot travel from one place to another through that layer or through that membrane. That membrane would never allow the entry and exit of particles through it. Not even a single particle can travel through it. Such layers or such membranes are referred as impermeable layers or impermeable membranes. Impermeable. No particle can enter, no particle can exit. Okay. Second one, this one, permeable membranes. Permeable membranes. Now permeable membrane means, okay, permeable membrane means there are like heavy gaps between the particle, between the material. Heavy gaps means each and every particle can travel here and there without any trouble at all. If you take water, we can dissolve like several substances within water, right? We can dissolve condense, we can dissolve salt, we can dissolve sugar, glucose, right? Then color pigments. You can dissolve like any kind of a substance within water, right? So my dear children, second one is the permeable membrane. There is no certain selection over there. Anyone can enter or exit very freely in and out from the membrane. Okay, anyone can enter, anyone can exit here and there through the membrane. Permeable membranes. It's like a polythene paper with large holes. Polythene paper with large holes. Okay. So if there is a polythene paper with large holes, any particle can enter through the holes and any particle inside the polythene bag can come outside through the holes. So these layers have, right, these layers or these permeable ones is going to allow the entry and exit of any kind of a particle. So any kind of particle can enter to the system through the permeable membrane. Any particle can exit the system through the permeable membrane. Then our one is this. So this is the this is the permeable one. Our one is the semi-permeable membrane. In a semi-permeable membrane, here by looking at the figure, you can get the idea. In a semi-permeable membrane, there are holes, but tiny holes. It's like a sand sifter. Sand sifter, you know. Now, when there is a construction of a house is going on, to make that cement plaster, to make some cement plasters, we have to use tiny sand, right? Without any pebbles. So in that case, we have to sift the sand. So that sand sifter is just like that. Okay, it's allowing to pass only smaller particles but not the larger ones. Larger particles remain within the sifter. Smaller ones are going to come out. So only selected particles can travel through it, but not all the particles. Such kind of membranes, such kind of layers are called as semi-permeable membranes. Semi-permeable membranes. Only selected ones can travel. Only selected ones can travel. Selected ones can travel. But however, if there is large particles or if the particles are not being selected, they can't travel. They remain within the container, they remain within the layer. So these kinds of ones are referred as semi-permeable membranes. Only selected ones can travel here and there. Okay, only selected ones can travel here and there. Semi-permeable membrane. So my dear children, once again, impermeable, permeable, semi-permeable. Impermeable means there are no any gaps, no one can't enter, no one can't exit. Permeable membrane, larger gaps, anyone can exit, anyone can enter. Semi-permeable membrane, tiny gaps. Only selected ones can enter. Only selected ones can exit. Semi-permeable membranes. Right. Then I am going to talk about the motion of the particles. So my dear children, you know that motion of the particles is going to happen or it's going to occur when there is a concentration difference only. In order to travel particles from one place to another, there should be a concentration there should be a concentration difference, like in the diffusion. So this is a special kind of a diffusion that's going to occur through a semi-permeable membrane, right? Diffusion which occurs through a semi-permeable membrane. In the earlier case, just the diffusion, there was no any other layer between particles. But however, in this process, there is a semi-permeable membrane which allowing only selected particles to travel. So my dear children, in order to travel particles, there should be a concentration difference, which means the particles are traveling upon a diffusion process. It is traveling under a diffusion, but through a semi-permeable membrane. So my dear children, osmosis is the diffusion process that's going to occur through a semi-permeable membrane. The diffusion which occurs through a semi-permeable membrane is referred as the osmosis. It's a special one. Unlike the diffusion, it's somewhat different. It's because there is a membrane between the particles right now. So membrane is allowing only a selected particle to travel. In the earlier case, each and every particle can travel here and there. Right? Each and every particle can travel here and there upon the concentration difference. But here, upon the concentration difference, there are only selected particles which can travel, others can't. Okay? We proved that by using the above experiment where we use condensed salt with water. Even though the concentration of condensed within the thistle funnel is very high, condensed particles never came to the beaker. Even though the salt concentration within the thistle funnel is very high, salt particles never came to the beaker. But however, water concentration was very high within the beaker, and within the thistle funnel it was very less. Only water entered to the system. Only water has the ability to travel through the cellophane paper. So this is a special kind of a diffusion process which is going to occur through a semi-permeable membrane. Right? So the diffusion that occurs through a semi-permeable membrane is referred as osmosis. A special kind of a diffusion process. Right then. So my dear children, osmosis, a semi-permeable membrane allows only some selected molecules to pass across it. Only selected molecules can pass across it, that's it. Only selected ones can pass across it. Okay, like water. You can do the same experiment using an egg membrane instead of the cellophane. So when you take an egg, right, there is a small bit of a layer between the outer crust of the eggshell and between the inner egg white, right? There is a small bit of a layer, egg membrane. So by using that membrane also, you can do the same experiment. Egg membrane is also going to behave as a semi-permeable membrane which allows to pass selected substances like water. Egg membrane is also considered as a semi-permeable membrane. So egg membrane is also a semi-permeable membrane. So that's why we can do this experiment, the above experiment, by using the egg membrane. So my dear children, you are given the process of osmosis. Let's see. Diffusion of water molecules. Especially here we are discussing about water, right? So diffusion of water molecules through a semi-permeable membrane from an area of higher water potential to an area of lower water potential is known as osmosis. So it's a process of diffusion, right? It's a process of diffusion. Diffusion in the sense means higher potential to a lower potential. Higher concentrated area to a lower concentrated area. That's the main idea of the diffusion, right? That's how the diffusion is going to work. But osmosis is a special kind of a process which is happening through only a semi-permeable membrane. It's only going to happen through a semi-permeable membrane. So the diffusion of water which is going to occur through a semi-permeable membrane is called as osmosis, right? Osmosis. So my dear children, we discussed that water is absorbed by root hairs and it is being transported from cell to cell. Within the cell, there is a special structure called a plasma membrane, cell membrane. Like the egg membrane, there is a membrane within a cell which is called as the plasma membrane, cell membrane. You know that we can draw out a cell like this, right? This is a typical cell. This outer layer is the one which is called as the cell membrane then. Right? Cell membrane. So water is coming towards, water inside the cell is coming out from this cell membrane through this cell membrane to the other cell. Like that way, from cell to cell, water is being traveled, water is getting transported. This method is osmosis. High concentrated area to a lower concentrated area through a semi-permeable membrane. Here the cell membrane or the plasma membrane of the cell membrane is also called as the plasma membrane, right? Plasma membrane. Plasma membrane. So here the water particles is going to travel through the plasma membrane or through the cell membrane from a higher concentrated area to a lower concentrated area. That's how the water is being transported from one particular cell in root hairs to the other until they meet up with the xylem tissue. Then xylem tissue is going to distribute water all around the plant. Right? So this is the process of osmosis. Special kind of a diffusion of water particles or water molecules. That's going to happen through a semi-permeable membrane. Here within cells, the cell membrane or the plasma membrane is the one which is behaving as a semi-permeable membrane. From a higher concentrated area to a lower concentrated area. Osmosis means that in the simple sense. Right? So always you know that diffusion means higher to a lower concentrated area. So diffusion of water molecules through a semi-permeable membrane is simply, we can say, osmosis. This is osmosis, my dear children. So I hope that you got a good knowledge about the osmotic process. Right. So to study about the osmosis further, let us do the assignment given. Now there is another assignment given in order to investigate further things related to the osmosis process. Right? So the second assignment, let's see. Take a petiole of a papaw leaf with a closed end. Now petiole should be like this. The bottom end should be closed. The one which is being fixed to the plant, it should be closed. There can't be any gaps in it. Okay? It should be a closed one. Special. Okay? Then fill it with a salt solution. Place it in a water container. Now we need to take this petiole and we need to insert some salt solution into it. We need to insert some salt solution. Right? We are inserting salt solution to increase the concentration of salt, but to reduce, right? And to reduce the concentration of water. So within the petiole, large number of water particles can be observed. Okay? Within the petiole, we can observe less number of water particles. Large number of sodium chloride or salt particles. As we are inserting salt solution into the petiole. Okay. So inside the petiole, large number of salt particles are there, but very less number of water, as we are inserting salt solution. Now within the beaker, however, there is large amount of water particles. So let me name these things to you guys. This is water and this is the salt solution. And this is the petiole, right? The three parts. So petiole is the one which is filled up with salt solution. Here beaker has water. Okay, this is a closed end. This is the part where it is being fixed to the plant. So this is a closed end. So we have to like very gently remove this petiole from the plant. Otherwise otherwise these areas would may contain gaps. If there are gaps, then we can't do this experiment. There should be any gaps. Remember. Okay. So let's see what's going to happen in it here. So now according to the figure, we need to place the petiole which contains the salt solution within the water beaker according to the figure. Then after that, we need to observe what's going to happen after few hours. So this is our experiment, my dear children. So I'm going to show you what's going to happen in here, right? Even though this is an assignment that you need to carry on in your house, I'm going to show you by doing this experiment in our laboratory what's going to happen. Before I'm going to conduct the activity, let's see what's going to happen over here. You know that this end is closed now. However, those things are made up with cells. Even though this end is closed, there are cells and cells contain cell membranes. Cell membranes allow water to pass. Inside the petiole, there is only salt solution. So the concentration of water, right? Let me rephrase. Concentration of water is very less within the petiole. Why is that? Because there is salt. However, the concentration of water is very high in the beaker, as there is only water. So automatically, water within the beaker will travel towards the petiole of the papo through the cells over here, cell membranes over here. So after some time, my dear children, definitely the water level should rise up through the petiole and it should pour down or pour out from the petiole. This should be our observation. Water should travel from beaker towards the petiole because the concentration difference is from here to here. Here high concentration, within the petiole low concentration because of the salt solution, right? As there are salt, water particles are less within the petiole. As there is only water within the beaker, concentration is high in the beaker. Means high number of water particles. So the particles will definitely travel, water particles will definitely travel through these cell membranes inside the papo tube. Right then, that should be our expected observation. So let's see whether this observation can be taken from our experiment or not. So let's head on to the lab, my dear children. We'll conduct this activity and let's see whether we can get that observation or not. Right, my dear children. To investigate further things related to the osmosis process, here I have used a petiole of a papaw plant, right? Papaw plant leaf. So that petiole is filled with a salt solution up to the topper level in here. Okay, it's filled up to the topmost level, a salt solution, right? Then this end of the petiole is closed. As you can see, it's been closed, right? So there is no way of entering or exiting particles from this end, right? As it is sealed, right? Now within the beaker you can observe water. Now inside the papaw petiole you can observe a salt solution, So if the particles are going to exchange between papaw petiole or with the solution or the water in here, there is only one way. That way is through this end, right? So let's see, we'll keep the system for a while and let's see what's going to happen with our system, right? So you know that if the papaw petiole contains salt solution, means that the water concentration within the tube, papaw tube, is very less. Water concentration is very less as it contains more salts. Within the beaker there is large number of water particles. So according to the osmosis process, what should happen here is water should come towards the papaw tube and it should drop off from this end to our board or to our desk in here. So let's see whether this thing is going to happen or not after few hours. So as you can see here, now the solution within the papaw tube has come to the very top and you can see that the water drop is coming down. See, there's a water drop coming down towards the papaw tube. You can observe it very clearly, right? So it concludes that water is going to draw from the beaker towards the papaw tube and it is coming away from this end. It's because of the osmosis process that's going to happen within the papaw tube. Papaw tube is allowing only water particles to enter to the system, right? However, the salt solution is not getting entered to the water in here. It's because of the difference between the number of particles. So water which is getting drawn into the papaw tube come towards this and it is getting expelled from the other end. So if you're going to keep this thing for a longer time, you can get a more closer observations. Right, my dear children. So as expected, you can clearly see that water is dripping off from the petiole because of traveling water from beaker to the petiole, right? So you could clearly observe that dripping off, right? So as we expected, as we have explained, right, the osmosis is going to work from a higher concentrated area to a lower concentrated area through semi-permeable membranes. Right, now let me write down our observation and conclusion, my dear children. So our observation was quite simple. What has happened in the observation? You could see that water is dripping off from the petiole, right? Water is going to drip off from the petiole of the papaw leaf. So you can write our observation. Water is Water is coming out from the top. Water is coming out from the top of the papopetiole So this is our observation. From the top of the papopetiole, water is coming out, right? Water is coming out from the top of the papopetiole. So conclusion. What is our final conclusion from the activity? Our final conclusion from the activity is quite simple. Water has traveled from beaker towards the papopetiole, right? So we can write water has traveled from the beaker to the papopetiole. Water has traveled from the beaker to the papopetiole. This is our conclusion, right? If you want, you can mention this thing as through osmosis. Through osmosis. Right. So this is our final conclusion, my dear children. Right. By the osmotic process, water has traveled from the beaker towards to the papopetiole. Right then. So my dear children, many minerals are dissolved in soil water. The root hairs of plants absorb these water particles by osmosis. Now, many minerals has been dissolved within soil water. You know that soil water contains many minerals. That's why it is referred as mineral water. Even the water that we are going to consume or that we are drinking, spring water, well water, those are mineral water. So we obtain this water from the soil. Soil has several minerals. So these minerals are dissolved in it. So these minerals, this water with minerals are referred as mineral water. So the root hairs of plants absorb these water particles by osmosis. Root hairs are going to absorb this water by the process of osmosis. I told you that cell membrane is the one which is behaving as the semi-permeable membrane, okay? Cell membrane or the plasma membrane. Then from root hair up to the xylem, water particles move from cell to cell by osmosis. So even I have explained this thing in the earlier case. From root hairs to the xylem, up to the xylem tissue, right? By one cell to another, water particles travel. This happens through the osmosis. Okay, it happens through the osmosis. Cell membrane is the one which is behaving as a semi-permeable membrane. Right, so the cell membrane inside the cell wall acts as the semi-permeable membrane. So here you are given with the figure, right? See, these are the soil particles. This is the root hair. Soil particles contains water. So that water is being absorbed by the root hair. Then root hair is going to pass that water into the several cells. Then from this cell to this one to this one, this one to this one, up to the xylem tissue. From cell to cell, water is being transported How does the water is getting transported? Through the process of osmosis. So within the cortex of these cells, from cell to another cell, right, by the process of osmosis, water is being transported up to the xylem tissue. Then xylem tissue is going to freely transport water within the plant here and there. Xylem is the tube which acts as the transportation of water, right? It's the transporter of water. It's the tube that's going to act as the transporter of water from throughout the plant, right? So, however, up to xylem tissue, water being transported from cell to another cell by the osmotic process, osmosis. Right, my dear children. So, I hope that you got a good knowledge about the osmosis, right? Then you are given with another topic, a subtopic, mass flow, right? Before I'm going to explain about the mass flow thing, see, root hairs absorb water by osmosis. Energy is not needed for this. Now there is a special process in osmosis plus as well as for the diffusion. For diffusion and for osmosis, energy is not needed. It's happening because of random movement of the particles. External energy is not being provided. We never provided external energy in each of these experiments, no. In each of those experiments which I did in our laboratory, I never gave any kind of a special force or special energy into those systems, right? Even though I'm not applying forces or even though I'm not applying any kind of a special energy, however, the expected observations could be observed, right? So energy was never needed for the processes of diffusion or for the osmosis. Special kind of an energy is not needed. So see here, but energy is needed to absorb minerals. Absolutely. Now, in order to absorb minerals from the plant, energy is needed. In order to transport food from leaf to other places through the phloem tissue, energy is needed. Right? Definitely energy is needed. So, for these processes, definitely energy is needed, my dear children. Those are not going to happen through the osmosis or diffusion. For osmosis and diffusion, energy is not needed. That's happening because of the concentration difference. Okay? Food produced in leaves are also transported to the other parts of the plant through the phloem tissues. So I told you that, right? Food which is being prepared within the plant leaf should transport it to different places. That should also happen by utilizing some amount of energy, right? There should be energy. Otherwise, these things can't transport from one place to another, no. Like how we can carry a heavy load of food substance from one place to another. We have to provide external energy. Plant should need or plant should get external energy for that process. Right? So my dear children, transportation of food from leaves to different parts of the plant through the phloem tissue is going to happen by utilizing some amount of energy. Energy is important in this process, in that transportation process. Then these photosynthetic products are transported as a unit along phloem tissues. And that process is known as mass flow. So my dear children, then photosynthetic products, these products like transportation of minerals and photosynthetic products like the transportation of sugars through phloem tissues, transportation of minerals, transportation of sugars. These things are very important. right? Not transportation of minerals, but absorption of minerals, right? So absorption of minerals and transportation of photosynthetic substances, like the transportation of sugars, right? I told you that starch is getting converted into a sugar solution if they are being transported to a different place through phloem tissue, right? So for this transportation process, some amount of energy is really important. So transportation of substances, especially here photosynthetic process, photosynthetic substances, right, is going to happen by utilizing energy within the plant. Actually, these things are transported. Photosynthetic products are transported through the plant by using a special kind of a tissue which is called as the phloem tissue. So to transport phloem tissue, to transport food within the phloem tissue, energy is important. So by utilizing energy as a single unit, these substances, these photosynthetic substances are transported along the phloem tissue. This process of transporting substances utilizing energy is referred as mass flow. This is called as the mass flow, right? This is the one which is referred as the mass flow, my dear children. So mass flow, in mass flow, utilizing energy, it is being transported. But in diffusion and osmosis, a different, way more different process. Here it is going to happen because of the concentration of the particles, okay? Mass flow is a different process. It is going to happen in transporting substances in the sense especially photosynthetic substances like sugars, starch, okay? In transporting these substances through the phloem tissue is utilizing energy within the plant. So this is referred as the mass flow, right? The third main type of transportation. So my dear children, we studied three main transportation processes that can be observed within a plant. Number one, the diffusion, which occurs because of the concentration difference, right? Diffusion means the random movement of particles from a higher concentrated area to a lower concentrated area. Then the osmosis. We conducted several experiments in this chapter to discuss about the osmosis. Osmosis is the diffusion which is going to occur through a semi-permeable membrane, right? Especially here we are discussing about diffusion of water particles. So diffusion of water particles which is going to occur through a semi-permeable membrane. This is called as the osmosis. Then the third one, mass flow. Mass flow is the absorption of, mass flow is going to happen during the absorption of minerals and transporting photosynthetic substances from one place to another through the phloem tissue. Mass flow. In mass flow, energy is being utilized, but however, during osmosis and diffusion, energy is being not utilized at all. So these are the three main transportation methods that we can observe within a plant leaf, my dear children. So here, once again, you are given with the methods of transportation in plants. The main methods of transportation in plants, my dear children. So we studied about three transportation methods mainly. Number one is the diffusion, right? Diffusion is the process which the particles move from higher concentrated area to lower concentrated area. Then the osmosis is the process where the diffusion of particles occurred through a semi-permeable membrane, and then the final one, the mass flow. In mass flow, energy is being utilized to transport substances from one place to another, especially in transporting photosynthetic substances like starch or sugars. The mass flow is being utilized by the phloem tissue. So these are the main transportation methods that we can observe within a plant. Main methods of transportation that we can observe within the plant. So my dear children, we finished up the lesson part, transportation. So these are the transportation methods that we can observe within the plant itself. So within our next chapter, we'll be discussing about other things related to the lesson, right? So we'll discuss other processes related to the plant in our next chapter.